So hello friends and my dear students. Today we are going to study the third experiment in the genetic material, as a DNA is a genetic material. The third experiment, Hershey and Chase experiment. After 7 years research work of the very tall experiment, after that the two scientists combinedly discovered Hershey and Chase experiment. Alfred Hershey and Martha Chase. They experimentally proved that DNA is a genetic materials. In this experiment, they used two important things. One is the bacteriophage. Bacteriophage means a virus containing DNA, and second, it is the bacteria Escherichia coli. It means these are the two important microorganisms he used in this experiment. Let us see how they performed the experiment, how they showed these two microorganisms play an important role in the DNA is a genetic material. So this is the practically they showed their experiment. In that experiment they selected two microorganisms. One is the bacteriophage and second one is the bacteria E. coli. So this is the first phase. This is the first phase where this is the bacteria. Within the bacteria there is the presence of bacterial chromosome. On the surface of cell wall of bacteria there is the attachment of phage. Phage means that is the bacteriophage, and there is the injection of phage chromosomes. This is the first process, first experimental stage, where there is the attachment of virus on the surface of cell wall of bacteria. That is the bacteria, which bacteria? E. coli. And during that there is only entry of DNA. Which DNA? Actually bacteriophage is made up of two parts. Bacteriophage is made up of only two parts. One is the DNA and second one is the protein. Only DNA part is entered into the bacterial cells. You just see. And whatever the protein parts, it remains outside the cell wall of bacteria. That's why this is the first stage. The first stage is called as attachment of phage. After that what happens? Only the DNA of bacteriophage entered into the bacterial cells, and once the bacteriophage DNA entered into the bacterial cells, what happens? There is the breakdown of bacterial chromosome. So why? There is the entry of virus DNA into the bacteria, but there is the breakdown of bacterial chromosome. Why? Because due to the effect of phage specific enzymes, and later on bacterial chromosomes totally broken down in the bacterial cells, and broken down chromosomes of bacteria, they becomes phage chromosomes. Why they becomes phage chromosomes? Because whatever the activity of DNA of bacteriophage entered into the bacterial chromosomes, and there is the replication of phage chromosomes using bacterial materials and phage enzymes. With the help of that there is the replication, and that's why the number of chromosome increases inside the bacterial cell wall. Later on, in the next stage, what happens? In the next stage, there is the expression of phage genes to produce phage structural components. Once the number of chromosome particles form in the last stage, in this stage what happens? Whatever the structural component of viruses or bacteriophages, they are produces within the bacterial cells, just like phage head, just like this is the phage sheath, base plate and tail fibers. These are the different parts of bacteriophage. Bacteriophage means viruses, and these parts are produces within the bacterial cells. How many numbers of these parts are produced? Thousands or much more numbers of these materials are produced within the bacterial cells. And in the next stage, what happens? In the next stage, assembly of progeny phage particles produces. Assembly means actually the virus or bacteriophage, it looks like head, tail, fibers, and within that there is the presence of DNA. That type of assembly they are produced within the bacterial cells. The number of such bacteriophage assemblies are also more, and like that the number increases, and due to the increases number, in the last stage what happens? These viruses, these bacteriophages comes out from the bacterial cells. Was how they comes out? Due to the increases the pressure of number of bacteriophages within the bacterial cells, due to the pressurized number of these bacteriophages within the bacteria, due to which what happens? There is the broken down, there is the breakage of cell wall of bacterial cells, and what happens? These bacteriophage they comes out from the bacterial cells. The numbers of releases of progeny phages by lysis of bacterial cells are much more, and further these bacteriophages are ready for attachment on cell wall of bacteria. Like that, this is the cycle, and this cycle is called as lytic cycle. This cycle is called as lytic cycle. And like that, what happens? There is the infection of such viruses, bacteriophages, to the host cells. In this experiment, host cell is a Escherichia coli. Not only host cell is Escherichia coli, but also if viruses attaches on human cells, plant cells, or any other cells, they work same, just like in this experiment the viruses attaches, entered, and after that there is the breakages. Like that the viruses they work, and the number of viruses increases, further they attaches to the host cells, and like that the number of viruses increases. So in this cycle, the Chase they said that in this way there is the increases number of viruses due to the entry of DNA of bacteriophage into the bacteria. After that, the Hershey and Chase experiment, again they carried out one experiment. So in this experiment they selected what? DNA with 3 to it means that they generally use what? They generally use two different radioactive compounds. One is the radioactive compound is the 32 phosphorus, and next one is the 35 sulfur. These are the labeled DNA with 32 phosphorus and labeled proteins with 35 sulfur. These are the radioactive compounds are selected in this experiment. Same in this experiment, what happens? In this experiment, this is the first batch in which within the conical flask there is the bacterial medium, there is the growth medium added, within which there is the addition of E. coli bacteria as well as bacteriophage, and there is the addition of radioactive sulfur. It means that this is in the conical flask there is the mixture of one growth medium, second bacteria, third bacteriophage, and number fourth, that is the radioactive sulfur. They are mixed, and after a period of times, after a incubation times, what happens? The such mixer or such culture, such mediums are afterwards added into the mixture or added into the blender for agitating the mixture, due to which what happens, there is the separation of phage outside the bacteria from the bacterial cells. It means that whatever the material present outside the bacterial cells, there is the presence of what? There is the presence of empty protein sheath. These are the empty protein parts of the bacteriophages. They are separated, and what remains? Only bacterial cells, within which there is the presence of DNA of bacteriophage. So afterwards what happens? There is the due to the centrifugation process, that centrifuged part, when we collected into the test tubes, the liquid parts collected on the upper, and at the bottom, that is the solid part. Solid part is called as pellets, and pellet, it is nothing but bacterial cells and its contents. And whatever the liquid parts upper side, that is nothing but it is a radioactivity. It means phage proteins in liquid forms. So this is the batch 1, where they perform or they proved that what? Only DNA entered into the bacterial cells, and whatever the protein parts of the bacteriophage, it again outside the bacterial cells, outside the, it means that separated in the liquid format. In the second stage, what happens? They use, so in the first stage what happens? Whatever they use radioactive sulfur, that sulfur is attaches to the protein part of the bacteriophage, not DNA parts. It means that sulfur is not responsible for transformations. It means that it is not the part of DNA. In the second stage, what happens? Same mixture, growth medium, viruses, bacteria, and in place of radioactive sulfur, there is the addition of radioactive phosphorus. Radioactive phosphorus is added. Radioactive, it is isotopic form, 32P. It is an isotopic form of phosphorus, generally added into the conical flask. So what happens? Whatever the radioactive compounds, that is phosphorus, it attaches to the DNA, because phosphorus, it is the part of DNA, and that's why the bacteriophage DNA, it becomes radioactive DNA. Radioactive DNA. Same process we again perform in this experiment, means there is the blending process. In the blending process, means in the mixing process, due to which whatever the outside of the bacterial cells, again they separated and remain what? In the bacterial cells, and within which there is the entry of DNA of bacteriophage, and it looks like green color. Why? Because radioactive phosphorus, it attaches to the DNA of bacteriophage DNA, and this is the what? Test tube. When we centrifuge that mixture, same liquid part present on the upper side and solid part remains again at the bottom. It is called as pellet. Pellet means that is the nothing but bacteria, within which the DNA of bacteriophages, and whatever remains, that liquid, it contains the protein part of the bacteriophage. And with the help of this experiment, clearly Harsh and Chase, They proved that DNA is a genetic material which is only responsible for transformations. So, and with the help of this, he indicates that he proved that what the DNA is transferred from bacteriophage to bacteria. That's why it is a transformation process occurred. It means that DNA is a genetic materials which transferred from one generation to another generation, and with the help of this experiment finally he proved that DNA is a genetic materials. So student and my dear friends, so with the end of this experiment I finally concluded that this experiment these three experiments proved DNA is a genetic materials. So friend, if you have any problems with respect to this experiment, so you just comment in the comments, that is in the YouTube comments box, and you ask if you have any problems regarding the experiment. I'm always ready for that. Till then, bye.